Okay, so we're going to start taking a look at biotechnology and genomics. We've actually been doing some genomics in class because of the journal club, but we're going to take a look at the procedures behind it and some of the things we've learned from genomics. So this little picture I liked because it sort of shows what we'd like to know when we look at cells and we look at human beings or bacteria or whatever. We know that the cells contain DNA and we want to understand that DNA, and of course genomics is all about sequencing the DNA, but part of the reason that we want to sequence the DNA is so that we know about the proteins that are encoded for by the DNA, and those proteins are little machines essentially that make this whole set of reactions that happens in a cell. This would be a big metabolic map showing all the reactions. Each of these little protein machines are essential to keeping these reactions going, keeping this cell living, and of course cells communicate with other cells, whether we're talking about bacteria living in a community or if we're talking about human cells that are gathered into a tissue. We also want to understand the interconnections between cells so that we understand how life relates and exchanges information to cause the body to function the way it does, to cause communities of bacteria to form biofilms, or for all the communities of different kinds of cells to exchange nutrients in a food web, right? Ultimately, we want to understand life, and DNA is at the base of all of that. And so we can work, and for years we have worked from the organisms and tried to work our way backwards. Genomics lets us dive into the DNA and from that code try and work our way forward. So it's very powerful, and it gives us another set of tools for trying to understand life. So this figure just reminds you of a couple of definitions that have come up before. A genome is all the DNA of an organism. The proteome is all the proteins of an organism. And in this lecture I'm focusing on genomes and genomics. This little graph shows the relationship between the number of protein coding genes and the total genome size, and you can see there is a very strong correlation. The bigger the genome, the more likely it is that, or the greater the number of protein encoding genes, right? Which probably is not a huge surprise. The other thing you can see on this figure is how the genome sizes stretch out. Eukaryotes are in green, prokaryotes in red, viruses in blue, and you can see that in general the genomes of eukaryotes are larger than those of prokaryotes, which are larger than those of viruses. We're out here somewhere. The units on this graph, I know it's really hard for you to see, but this genome size is expressed here in megabase pairs. So DNA size is measured in length of the DNA molecules, and length is counted by base pairs. So if you think of the rungs of the twisted ladder, right, each rung of the ladder is a base pair. So the longer, the more bases, the more base pairs in a molecule of DNA, the bigger it is. And when we're talking about genomes, we're getting into such huge sizes that we're not going to talk about base pairs. We're not even going to talk about kilobase pairs, which are thousands of base pairs. We're going to talk about megabase pairs, which are millions of base pairs. And the human genome, if we look at the human genome, the haploid genome, so just 23 chromosomes, was measured out at about 3 billion base pairs. So this axis here, this is times 10 to the 2 for megabase pairs, so 1,000 megabase pairs, so 10 to the 3 would be a billion base pairs, and so that's out here. So human genome size is right out in here. And then, you know, moving down into the prokaryotes, here this would be about 1 megabase pair, so 1 million base pairs. So you can see prokaryotes are, you know, a little higher than that. And then viruses getting very, very, very small, down to just thousands of base pairs in some viral genomes. So I used this slide once before when I kind of gave my introductory CommGen talk, and I said genomic analysis is like having a code, right? So we can read the code of the DNA, we can know the A's, T's, C's, and G's, but we don't really know what it all means. It's like hieroglyphics, and we have to figure it out. And the way we figured out hieroglyphics was with the Rosetta Stone, because it had hieroglyphics and ancient Greek on it, and we were able to decode it. So genomic analysis is like that. We get those codes, those A's, C's, G's, and T's, and then we have to figure out what it means. And some of the ways we can figure out what it means is by looking into databases of genes that people have already figured out. So if you have a sequence and you think, wow, what does this one mean? You can type it into the database and see if anybody has identified a protein that comes from that gene before and maybe figured out what it does. Sometimes we can just do comparative studies between sequences. We might not know exactly what the sequence codes for, but we could still look at similarities between organisms, right? So that's another way to sort of get meaning out of genomes. And if we look at the kinds of different things that people do with genomic analysis, the purpose of it ultimately is to determine the DNA sequence of the entire genome, right? But why would we want to do that? Well, we may be exploring functional genomics, where we want to figure out how many genes are in there. Can we find the genes that are located in that genome? And remember that protein encoding genes are going to start with, in the mRNA, it's going to start with a start codon, AUG, and it's going to end with a stop codon. And we can find start and stop codons in the DNA, right? Because the mRNA comes from the DNA. So we can look in the DNA and we can find those open reading frames and say, here's a gene. Okay, what's the sequence? And then try to figure out what is the function of that gene, what protein comes from the gene, what job does it do in the cell. And so that's functional genomics. And then, of course, there's this whole field of what's called metagenomics. And this is something I've talked about in class, although I haven't used that word. But this is when you go into an environment, and that would include the environment of the human body, and you just find out what genomes are there. So I've mentioned before that you really can't grow a lot of microbes in the lab. Less than 1% of the bacteria that are out there or on us will grow in the lab. But now that we have genomics, we don't have to grow everything in the lab in order to study it, at least to some level. Ultimately, you do want to try and follow up genomics with other approaches, but initially I can just take the DNA out of the microbes in the gut, and I can sequence their genomes, and I can compare them to known microbes and get a profile of what's living in the gut without ever having grown them in the lab, right? So that's called metagenomics, is just looking at these genomes from the environment. We can also do comparative genomics. We can say, what's the relationship between organisms? And very frequently, this is done by comparing the genes for ribosomal RNA. So ribosomal RNA makes up the ribosome. All cells have ribosomes, so it's a good molecule for comparing across a wide variety of living things, all the way from bacteria to humans. Everybody's got ribosomes, so we can compare that. So we can figure out sort of the evolutionary history of life on Earth, and we can explore evolutionary questions. We can look at coevolution between microbes and their hosts, right? We can ask other sorts of questions that have an evolutionary basis. And then, of course, there's this whole idea of personalized medicine. So we are all different. Humans are all different from each other. And so we have certain proteins that we make that might be drug targets. And maybe there's one drug that's going to work perfectly on one version of the protein and a different drug that'll work on a different version of the protein. So if you and I both need that treatment, whatever it is, and I'm talking about in the future now, we're not quite there yet, we could sequence our DNA code for that particular protein, see which variant we have, and then prescribe the drug that works best for that variant. So the more we know about our individual differences, the more we can look for ways to tailor medicine towards those differences, right? We can also look at our genomes and say, what's your disease risk? So you can, in the future, again, we may take the human genome, having your DNA sequence might be part of your medical history, and look for risk factors in your genome. If you knew you had a slightly higher risk of a certain kind of cancer, you might choose to eat differently, exercise more, right? You might base your lifestyle to minimize your risks. So maybe in the future, although with all of this, there are definite ethical implications that come from having this sorts of knowledge. Who would have the knowledge? Would your insurance company have the knowledge, right? So there's lots of ethical things that need to be considered, and they are. There's conversations happening about those sorts of things, but these are sort of the potentials of where we might go in medicine with genomics. And so I already mentioned this one about which drugs will work best. Okay? And that idea of tailoring drugs to a person's genome is a field called pharmacogenomics. So if we take a look at what's happened in genomics, and this is a very young field, right? We didn't really get the ability to work with entire genomes until the 90s. The Human Genome Project started in 1990, and back then this is what the facility looked like. This was the many different computers. It took months, years to get a whole genome sequenced, and nowadays there are personal desktop sequencers. This is the MeSeq desktop sequencer that's made by Illumina. So you could have your own personal little DNA sequencer that just sits on a tabletop. Much smaller, more compact, and much faster. In as little as 14 minutes, you could have a genome of something small, right? Not a big genome, but a small genome in as little as 14 minutes. So we've come a long way, baby. The other thing that's come down a lot is the cost of getting a genome, right? So here we are again back at the beginning of the Human Genome Project, right? Millions of dollars to sequence a genome. Look at this. It's coming down, and the cost is just dropping and dropping, which makes it more available. So instead of having to have these huge government facilities or these enormous corporations that are doing genomics, individual researchers, individual scientists can get grants that are big enough to buy one of those small sequencers. More labs are developing the ability to get genomic data, and so that's really great. It's expanding, and opportunities are opening up because of the drop in cost. Right? So what we're going to do in the next couple of lectures is we're going to look at a few of the key pieces of biotechnology procedures that underlie genomic research, and in particular we're going to focus on the techniques that we're going to be using in the ComGen project. We're going to look at how we make genomic libraries, gel electrophoresis, which I think probably you're familiar with from cell biology, polymerase chain reaction, and the DNA sequencing process. And I'm not really going to talk about the computing aspects of all this, but just imagine the vast amounts of information that are flowing in to computer databases because of genomics, right? So all of these, billions of base pairs of DNA being sequenced out there, and if more labs are able to have their own sequencers, that means there's more data being collected. That's just a ton of information. I mean, imagine libraries filled with books that need to be read, right? So because of that, there is a really big need for computing to go along with all of this information, because we have these databases and we need to be able to analyze them using statistics and figure out what does it all mean. There's actually a whole new field of biology that wasn't even born until after the genomic sort of revolution, and that field is called bioinformatics, and this is a field that kind of melds computer science with biology, and it's all of the tools that are needed to manage this information, to kind of analyze this information. So there's just a whole new field that was created because of the need to deal with this data. Okay, so that's kind of the intro of where we're going, and I'm going to pick up in the next clip with talking about how you make a genomic library.